Hi, welcome to this small lecture about structural biology in solution. So, structural biology is a branch of science that deals with characterization of individual constituents of a macromolecule in three-dimensional space by biophysical methods. So, the macromolecule, like shown here, DNA-K, a protein molecule, consists of 638 amino acid residues. So, biophysical characterization of the individual amino acids in three-dimensional space. For example, the amino acids can be arranged in alpha helical structure, beta sheet structure, or they may be structureless in random coil, and then their overall arrangement with respect to each other in domains, like here it is in NBD, SPD, lid, and their certain arrangement in the space is what structural biology does, is what structural biology tells us about the molecule, and this is attained by certain biophysical and bioanalytical techniques. So the obvious question is, why do we need to know structural biology? Because the information that we get from structural biology contributes to understanding our understanding of the molecule towards the metabolism, biochemistry, and biology that's happening inside the cell. For example, again, I want to show you here DNA-K arrangement of its domains in the three-dimensional space. Now, if a substrate comes in and binds to this molecule, you see its molecular structure changes to a huge extent, to a large extent. Its lid domain moves around 90 degrees and clamps on the substrate binding domain. Now, structural biology tells us about what structural changes drive a reaction, or what structures are attained as a result of a certain biological reaction. Why do we need to know again these structural changes? Next question would be, why do we need to know the structural changes at all? The answer for that is that once we know the structure, we are in a better position to devise a drug that can inhibit or enhance the activity of such molecules. And that's very important, because if we consider example of this molecule, that's one of the heat shock proteins, HSP70, it's implicated in cancer. Now, if we have a molecule that inhibits its activity, then the biological activity of this molecule will be stopped, and as a result, the cell function, cell metabolism will be altered. Now, if we have an inhibitor of this molecule that can inhibit the function of this molecule, that will directly result in the stoppage of the cancerous carcinogenic cells. So having information about about molecule from structural biology contributes directly to the disease management. So what are the techniques by which we gain information about structure of the molecule? X-ray is one of the mostly trusted ultimate technique that gives us information about the three-dimensional orientation of the molecule, or three-dimensional structure of the molecule. Now, the limitations of X-ray is that it gives us a static information. It does not give us a dynamic information of the molecule. For getting dynamic information of the molecule, we use other set of techniques in solution. That's NMR, ESR, fluorescence, CD, and FTIR. EM, to a certain extent, is considered dynamic. Now, for the purpose of this lecture, I will only talk about fluorescence. So students who have some information about biochemistry and have a little information about analytical chemistry basically will be able to follow the lecture quite easily. So fluorescence has been reported from 1852 by George Gabriel Stokes while he was working on fluorspar and uranium. And these seemed to him to convert UV radiations into longer visible radiations. The principle of fluorescence can be described as the phenomena where a molecule absorbs light and emits it in visible range. Pictorially, we can explain that in this fashion. For example, we consider a molecule that has a fluorescent molecule. We consider big, large macromolecule that has a fluorescent moiety inside it. Not every molecule is fluorescent. We'll talk about that later. Now, if we shine light on it and we are able to record its absorption, we will see that the light will be absorbed at certain wavelength. Now, if we have a condition where we are able to completely take this out, the incident light, and monitor the emitted light from this molecule, we will see that emits fluorescence at a wavelength higher than the wavelength where it absorbed, and consequently this will be of lower energy than where it absorbed. On the electronic scale, the electronic process of this fluorescence is explained on the basis of Jablonski diagram that says that when the electron is in ground state and it is shined, it takes a photon of energy in the form of light, and it gets excited to an excited state from where it starts losing energy through internal conversion, and when it reaches the lowest possible energy level in the excited state, it jumps to the ground state, emitting energy in the form of fluorescence. This diagram also explains the difference between the energy of absorption and energy of emitted fluorescence light. The process is very fast. It takes only 10 raised to minus 15 seconds for the fluorescence to occur. So the accepted rules for excitation, there are many rules. One of the rules is Franck-Cordon principle, which states that at one time only one electron will be excited from the ground state, and it will be excited to a symmetrical energy level. That means if the electron is residing in pi, it goes to pi star. If it's in sigma, it goes to sigma star. People or students who have a better grasp of molecular orbital theory will understand what I'm talking about here. So there are many things about fluorescence, but I will not talk about these. I will only like to mention here that fluorescence is a dynamic technique, and it's used in a vast number of fields. It's used in knowing the flexibility of the molecule by polarization and anisotropy. Quenching gives us the accessibility of the fluorophore in a molecule. Fluorescence resonance energy transfer is used for measuring distance between various domains. In cell biology, fluorescence assisted cell sorting helps. And again, localization of macromolecules inside the living cell can be also done by confocal microscopy. And as I said earlier, not every molecule exhibits fluorescence. There are only few molecules, mostly they are aromatic molecules, that are able to absorb energy and then release it in the form of fluorescence. These are some examples of those molecules. Psi3 and Psi5 are the dyes that are mostly used in nucleotide sequencing. And recently, for the last 10 years or so, green fluorescent proteins have evolved, and they have been used exhaustively in knowing the localization of the molecules in the cells, in knowing about structure and dynamics of the molecules, and in knowing about mechanisms of the protein-protein interactions in general. So I will not talk about the source and setup of the instrumentation. I will directly go to the use of fluorescence in the study of structure of a protein that's a macromolecule. Now, there are two methods by which we can measure the changes in the structure of protein. One is intrinsic fluorescence, and the other is extrinsic fluorescence. Intrinsic fluorescence is the fluorescence endowed to a protein molecule because of its constituent amino acids like tryptophan, tyrosine, phenylalanine that are fluorescent. They absorb at certain wavelength, they emit at certain wavelength, they emit fluorescence at certain wavelength, and they have good, for example, tryptophan has got a good quantum yield. So also FAD and FMN, which are part as cofactors of the protein, also endo a protein with an intrinsic fluorescence. Now, if we are able to follow the intrinsic changes in the intrinsic fluorescence, we will be able to tell what dynamic structural changes are taking place in a protein. And I have tried to catch it show it in a pictorial form. Here we can see that a protein which has a fluorescent moiety inside, usually in the native state it's folded, and the fluorescent moiety usually lies buried inside the hydrophobic environment of the protein. It's solvent inaccessible. There is limited access of solvent to this fluorophore, and the fluorescence is usually quenched in this state. That results in its emission maxima of 325 nanometers. Now, if we are able to unfold this protein with denaturation, with heat denaturation or guanidine hydrochloride denaturation, we see that the fluorophore becomes exposed to the environment. And when it becomes exposed to the environment, it becomes solvent accessible. Now, what happens to the fluorescent properties of this moiety is they change. They change. Why do they change? Again, we go to this Jablonski diagram. According to molecular orbital theory, the anti-binding orbitals are very polar, and thus they are very sensitive to the changes in the environment. So when the environment changes, the fluorescent properties of this molecule change, and consequently it emits at a very higher wavelength, lower energy. So now if we have an unknown sample and we are able to measure its fluorescence, we can directly say whether it's in a folded or unfolded form. And an example of this is directly from my data, where we had treated cytochrome C, taken as a protein, and treated it with trichloroacetic acid and trifluoroacetic acid, and want to see what structural changes are taking place by increasing the concentration of acid. We can see when there is no acid present, the relative fluorescence intensity of the molecule is somewhere here. But when we increase the concentration of acid, the fluorescence intensity of tryptophan, in this case, increases, showing that there is a change in the environment around this tryptophan residue. And if we go further increasing the acid, the fluorescence changes into a different, the environment of tryptophan is intensity changes of this tryptophan, directly telling us that the environment of tryptophan at this stage is different. Now we can divide this whole spectrum into three regions, 1, 2, and 3, and say that tryptophan is largely in one environment here, it's largely in another environment here, it's largely in a different environment here. So the three different environments of the protein of the tryptophan moiety with the increasing concentration of the salt is evident from this technique. But that's what fluorescence gives us, and that's what the information fluorescence gives us. Obviously, in this case, we have to use another techniques to support our claims, and in this case, that was done by circular dichroism, and it was found that the alpha helical and beta sheet content of the protein changed, altered, similar to what we were getting in the fluorescence pattern. Thus, fluorescence was able to, this information, what we gain from fluorescence, we cannot get it from X-ray technique. So fluorescence in this way gives us a lot of structural and dynamic changes taking place in the protein under these conditions. Another example shown here is when we monitor tryptophan emission maxima, and we see, for example, for this protein, FPA, when we treat it with guanidine hydrochloride, we don't see any changes in the lambda max of fluorescence up to close to 1 molar guanidine hydrochloride, from where it starts changing, and then again it becomes static. There are no changes in that emission maxima. So according to, again, our concept, as we showed here, in this case, the protein, in this case, below 1 millimolar, 1 molar guanidine hydrochloride is largely folded. It's largely unfolded above 3 molar concentrations of guanidine hydrochloride, and between 3 and 1, it exists in different intermediate states. Again, in this paper, they have proved that with another technique like CD, and I think they have used some other techniques to show that this result corroborates with what they find in other techniques. So to add to it, the data obtained in such fashion can also be used by applying certain equations on it. We can get free energy of the system, we can get the kinetics of unfolding and folding reactions. Again, this information is very useful for knowing the system, knowing its contribution to the metabolism of the cell in general. The second concept that is used for knowing the structure of protein through fluorescence is extrinsic fluorescence. In extrinsic fluorescence, fluorophores that show fluorescence are added to the system from outside, but they can bind to the protein, and after binding to the protein, their fluorescence properties changes. But the one fundamental portion of that is that they bind protein in a different structural form. For example, here is ANS, it does not bind to the protein in the folded form, but when it's unfolded, it binds to the hydrophobic patches. So if we monitor ANS fluorescence of a system with respect to time or with respect to denaturant concentration, we can see that it does not bind initially, indicating that the protein is folded. And after a certain point, when it starts binding, that means that the protein has started unfolding. It has started exposing its hydrophobic patches, so that the external fluorophore that was there is binding to the molecule and changing its fluorescent properties. So in this way, we can monitor the folding and unfolding of a protein in the real time. So again, as similar curves and information can be obtained from extrinsic fluorophores, as I showed in the intrinsic fluorophores. Thus, fluorescence can provide huge information about structure and dynamics of the molecule in solution that X-ray cannot, and this information can be useful for drug design. I cannot go into the details in this short format and lecture, but there's a whole lot of information that we can get from structural dynamics of the molecule that are very useful. Thank you very much for your patience.